All right, this week we're talking about the cell and looking at the structure and function of cells. So we're going to talk about prokaryotic cells briefly. However, we don't spend a lot of time on prokaryotic cells. We spend more time on eukaryotic cells in this particular class, but we will touch on what they are so you know the difference between the two. So a prokaryote has no membrane-bound nucleus. So the nucleus usually sits in the center of the cell, and it is surrounded by a plasma membrane that protects it. In prokaryotes, there is no membrane around it. So think of no pro. They're kind of opposites, but that remind you of how to kind of rhyme it. Basically, all their nuclear material, so all their DNA, kind of is housed in a region, an area, kind of throughout the cell. So they are very simple structures. Usually they are unicellular, meaning that they're a one-celled organisms. And prokaryotic cells fall under two domains. They fall under the bacteria domain, which is primarily what we think of when we think of prokaryotic cells. And they also fall under the domain archaea. And remember, archaea are extreme environments, unicellular organisms that usually are aquatic. The domain bacteria is quite large. They don't necessarily have to live in extreme environments. They could be in water, aqueous, but they don't need to be. So here's an example of what a prokaryotic cell might look like. So on the inside, you'll see this is our DNA, the nucleoid, nucleus material. It is in what we call a region. It has no outside capsule protecting it. You will see also there's obviously some cytoplasm, so liquid material, and there are a few other structures that we consider are small organelles. Organelles are small structures that have a specific function and a job within the cell to help keep the cell alive. And you'll see that everything's kind of free-floating here. Now, what you will notice about these wonderful prokaryotes is they have three layers of protection around the outside of the organism. So they have a plasma membrane, and then on top of that, they have a thick cell wall. And on top of that, they have an even thicker capsule wall. So they have three layers that protect them from the outside. However, once something gets through all three of these layers, then it can definitely disrupt easily the DNA on the inside. You will also find that these particular cells have what we call cell modifications, which means that they have some type of structure on the outside of the capsule. Here we have a long flagella, which is a tail. These are what they call pili. They almost look like little tiny hair projections coming off. So there's an example of them. Each one of these little kind of cell modifications help to keep this particular organism having the ability to locomote, so be able to move around. Now, so this is an example of a rod-shaped bacteria. Remember, we need to get through these three cells in order to destroy bacteria. So here again, our rod-shaped bacilla is the name we might see when we're looking at them. These guys are kind of spherical. Some are circular, some are oval, and we call those cocci. And then we can have these other ones that kind of look like a corkscrew or like the cord of a telephone. Think about an old landline telephone, that is, a corkscrew, and they are what we call spiral. So there's some different types of bacteria that we would see. Now, we don't spend much time on bacteria in this class. That is a whole entirely separate class called microbiology. Sometimes in 108 we'll spend a little bit of time on bacteria, but for the most part we don't spend a lot of time on prokaryotes in this particular course. We spend most of our time on eukaryote cells. Now we fall under that big domain of eukarya, and these are more familiar organisms to us. Now, what's the difference? They have a nucleus, just like prokaryote cells. However, the nucleus has this large protective cover around it that protects the DNA on the inside. We also have quite a few different organelles. So again, those are those specialized little structures that have a defined function. So our cells have a lot more of them, and they have specific functions to help keep the cell alive. Now, eukaryote cells can be unicellular, so one cell, or they can be multicellular, many-celled organisms. Such as humans, we are multicellular. So eukaryotic cells have a membrane-bound nucleus, and the other thing that we like to do inside of our wonderful cells is we have this compartmentalization, meaning that it's very organized. We kind of separate those organizational skills that our cells might have into what we call the endomembrane systems, meaning membranes within the cells that can kind of transport and communicate and move things. And then we have these little organelles that are energy related. So their job is to basically create energy for the cells. So they're pretty self-sufficient and they're very independent of any other organelles in the actual cell itself. So if we look at eukaryote, we can have animal cells, we can have plant cells, we can have fungi cells. We have lots of different types of cells we can talk about here. This is a typical animal cell example. You can see in the middle here we have a nucleus with our nucleolus, which has all of our DNA. And on the outside we kind of have this orange structure, which we call the plasma membrane. That is protecting all our precious DNA material and our protein-based amino acids that are being built here with our RNA. So this is very much protected. Now what you'll notice is, again, we have little structures that are compartmentalized and they all have specific jobs. And on the outside, our exterior surface is what we call a plasma membrane. We don't have any other kind of outside protective layer like that wonderful prokaryote does. We just had one of those layers. And that gives good flexibility, but it also can be a challenge for some of our cells at certain times. Plant cells are another type of eukaryote cells, and this is another type that we would look at primarily throughout this class. You'll see here that, again, it has a nucleus with a membrane. We have some of those very similar organelles. However, we do have a couple of different organelles here. We have a chloroplast, we have a vacuole. So these are different. We don't have these big structures pretty much in our animal cells. And the other thing you'll notice on the outside is that we have a plasma membrane. It's a little hard to see, but it kind of runs along the inside here. But these plant cells do have a cell wall on the outside. So plants have two layers on the external portion that keep it protected from foreign objects. Now, both of those, all three of those types of cells have what we call the plasma membrane. Now, a plasma membrane is really important. It's going to be made up of some specific molecules. It's what we call a phospholipid bilayer. A phospho is made up of a phosphate, a glycerin molecule, and those are what are represented as kind of these orange little circles. And each one of these orange little circles kind of have these two tails that they look like a V in this picture. And those are the lipids. Now, lipids are types of fats. And when we call this a bilayer, that means that I have one layer here on the bottom, and I have one layer on the top. So here's my one layer on the bottom. And then I have another layer on the top. So bi meaning two layers. Now, what you'll notice is that the tails both face the inside, okay? They're facing each other. And that is because these guys are what we call hydrophobic, meaning they do not like water. They are not water lovers. So think about that. Water mixed with lipids, so it's some type of oil or fat, don't mix. And we know that in our everyday life. So they sit on the inside, where these glycerin, so a phosphate and glycerin, orange little kind of molecules, sit on the outside of the layer. That could be the outside of the cell, or it could be the inside of the cell. And what we know about the outside of the cell and the inside of the cell is there's lots of H2O substances. So there's a lot of water here, and there's a lot of water here. So that wouldn't work if our lipids were facing that other direction. So this plasma membrane is going to surround our cells, our organelles, our nucleus. So a lot of those wonderful structures. Now we call this semi-permeable. So semi-permeable, let me see if I can write that real quick for you guys. And that means that it has the ability, and I think I missed a vowel there, has the ability to allow certain things into and other things out of the cell based on their shape, based on how large of a molecule they may be, and based on their polarity, so their charge. But within this, we might have channels, we have proteins. We have integrated proteins, which sit on the inside throughout the cell plasma membrane. We might have surface proteins, which sit on the surface near all the glycerol and phosphate molecules. Some of these that are integrated might be channels, so things can move into and out of. And they're kind of pathways to let things move. Now, more things that move through these channels are larger in shape and size than other structures. So let's talk about these organelles. The central kind of command center of all of our cells is the nucleus. It houses all of the DNA. It's very, very pertinent that our nucleus stays intact and that nothing invades it. So therefore, it has what we call a nuclear envelope. A nuclear envelope is a plasma membrane. It just has a fancier name that we call it nuclear. Oops, membrane. Sorry. We just call it specifically a nuclear envelope because it's around the nucleus. An envelope kind of encases it. Now, one thing about this plasma membrane is it does have pores. So not necessarily channels, it just has little tiny openings. So a pore might just look like a little tiny little opening just like this. And again, that allows things to go in and out that are of importance. Now inside the small center, so this is our nucleus here, let's say, inside the nucleus we have a smaller organelle, this guy right here, and that is what we call the nucleolus. And so that's this dark nucleolus that's in the middle, and that's composed primarily of RNA. And now RNA is going to help us decide and make certain types of proteins. But around that, we're also going to have this kind of chromatin, it looks like all squiggled rolled up stuff, and that kind of is our DNA. So sorry, I am not an artist. I am scribbling like there's no tomorrow. So that every cell is going to have a nucleus. So here's an example of what it actually looks like. Here's that nuclear envelope. You can see it has tiny little holes in it because those are the pores that allow things to move in and out. Here's the nucleolus in the middle where we have all of that kind of RNA and DNA structures. Now this is very important to the cell, and so therefore it's going to sit usually, let me go back, centrally located. So here it is kind of in the center. In our plant cell, it's a little off to the side, but that is pretty much the central kind of the brain of the cell, so to say. Now I said we wanted to make proteins, so what helps us make proteins is, yes, our RNA, okay, which is coming from inside the nucleolus. Whoops. Whoa, nucleolus, sorry, bad handwriting there. That's what we're going to use here. RNA is going to basically compose our ribosomes, and the ribosomes then are going to make proteins. Proteins are really, really important for our body, for our cells. So ribosomes are going to make the proteins. Ribosomes are tiny little specks. They kind of just look like little polka dots around. They might be free floating in the cell, or they might be attached to something, and then they're what we call studded or embedded. So if they're in the cytoplasm, they are free, okay? So if they're free, they're just kind of floating around. Or they might be bound to something. So if this is like our ER, we're going to see them kind of attached to them, and here they would be bound on the endoplasmic reticulum. And I'll show you a picture of them as well. So some of this endomembrane system, again, are compartmentalized, oh my goodness, I can't say that word right now. Compartmentalization usually means that they need some kind of enzymatic reaction. Now, enzymes help break things down so they can be easily worked with. So we have our endoplasmic reticulum. The endoplasmic reticulum sometimes is just referred to as ER. All right, we do have our Golgi apparatus, which has these little vesicles. Vesicles are tiny little structures that are transporting materials. Then we have lysosomes we'll talk about, and then we do have some vacuoles in our cell as animals, but they're not very big. In plant cells, they are quite large. So let's start with this ER. So you'll see I've written ER here. We have two types. The first type is rough. The second type is smooth. Rough ER is rough because it's studded with ribosomes. So this is, again, those little ribosomes that are sitting on there. And so basically the job of the rough ER is to synthesize. Synthesize basically means make or produce proteins, because again, what's going to help produce these, the ribosomes are, or it's going to modify, and modify means to change something. So I'm going to change proteins. Smooth has no ribosomes, and so what it synthesizes is lipids. So remember, lipids are a type of fat. Okay, ribosomes are going to work with the proteins. So rough ER, also known as RER, is going to do protein synthesis. Smooth ER, which is SER, sometimes has to do with the lipids. So I will show you what these guys look like. So here's our nucleus. Attached to the nucleus, we are going to find the RER, the rough ER, and that is again because ribosomes are being made inside the nucleus. They're going to come out, they're attached here, so protein. Next to that, you will find the SER, which is multiple around the cell throughout. It is smooth in appearance and texture.